It's a great pleasure for me to introduce today's seminar speaker, Ana Janic, who is a postdoc in my lab since about 9 months now. So Ana did her undergraduate in Belgrade in Serbia, and she then did some stints of getting further education in several labs in Europe before she settled in the University of Barcelona to study in fly genetics and study what is still something that we don't know enough about here in our opinion, and that is to use the fly as a model for cancer research rather than using the mouse or the human. So Ana is, in my opinion, a very brave young scientist to change from having been very successful in fly genetics to go into a lab that works exclusively with mice. I think that's a very big step, and she has a very questioning mind. She's very interested in many things we do, and therefore she's the ideal postdoc for my lab. Almost ideal. She doesn't play soccer, but her husband makes up for it fantastically. So thank you, Ana, for telling us about what we should know more about here, and that is an introduction into how to use the fly for cancer research, and the title of her talk is Germ Cell Genes in Cancer.
So thank you, Andreas, for a very kind introduction. So as Andreas already told you, so I was studying flies as a model for studying tumor genesis during my PhD. And today I will tell you about my main project, which I did. So first of all, I will give you a small introduction about Drosophila tumors. So Drosophila tumors have not been naturally occurring tumors in nature, so until now have not been reported. But Drosophila tumors can be actually experimentally induced by the mutations in some specific genes. And most of those genes have been identified in a screen for the recessive lethal mutations that caused the in situ lethal growth in 4 different types of the tissue in Drosophila. So it's in imaginal discs, in brain, in blood cells, and also in gonads. So the Drosophila tumors can be divided in hyperplastic and neoplastic tumors. Hyperplastic tumors are the tumors which have potential to overproliferate, but eventually they can stop and then they can undergo terminal differentiation, and also they are not invasive. While the neoplastic tumors, what happens, they are highly malignant and they are invasive and they are over-proliferating without the limits. So here in this picture you can see that there is just a representative example of the larval imaginal disc in Drosophila, which you can see the mutant and here the wild type, and this is just a confocal section which is showing that there is completely loss of organization of the tissue, as for example here we have a very nice multi-layer in the wild type disc. Also, you can see here in Drosophila brain what happens is that the optic globes compared to the wild type optic globes are much bigger, so they overproliferate. And also you can see here in phosphohistone in red is the mitotic index, so they have a higher mitotic index than compared to the wild type brains. Now there are dozens of genes that are known in Drosophila to be known as a tumor suppressor or the oncogenes, and they are involved in many important pathways that are important for the mammalian tumorigenesis, like Notch pathway, Bingless pathway, which is the Wnt pathway in the mammalians, also a DPP pathway, YAG-STAT pathway, or retinoblastoma, and many more. So one of the golden standard assay, how we assay the tumor growth in Drosophila, is what we do basically is that we label the brain tissue in this case, but this can be also labeled the other types of the tissue in Drosophila. We label with a GFP. Then we take this tissue from the larvae, dissect, and cut into the pieces. Then we take the very small needles and inject into the abdomen of the fly. So this is just a normal wild-type fly, which is around a 1-week-old fly. And then we implant these pieces of the tissue. If these pieces of the tissue is non-tumorigenic, then it will stay inside of the abdomen of the fly and eventually disappear. And if this tissue is tumorigenic, what will happen, it will just fill the abdomen of the fly and eventually give rise to the small micrometastases. And this can be repeated many times, so meaning that we can just retransplant this tissue many times. This is just a real example of what I have shown you in this previous cartoon. As you can see here, we have the pieces of, with the black arrow is just the place where we inject the tissue. And you can see here the wild-type tissue, which stays and never proliferates, eventually disappears. And this fly can live as a normal fly 4 or 5 weeks. But as you can see here with a tumoral implant, what happens, it fills the abdominal cavity of the fly and eventually can metastasize and form the small micrometastases in the thorax and also in the eye. Also in these sagittal sections of with a DAPI staining through the fly, as you can see here, we have the it's shown how the tumor overcome the complete abdomen of the fly. As you can see here, we have only one mature oocyte compared to the wild-type fly, which have many mature oocytes, and also ovarial, which here they are completely disappeared, and also the small micrometastases. So with this one here, I just want to tell you that this, so the Drosophila is much more different than the human tumors. So, but still we have some characteristic we can follow in this model, which can be quite interesting, which is that they are basically lethal to the host, because this fly will die in some cases of 2 weeks, but if the tumor is really aggressive, this fly can die just in a few days. And also, as you can see, they are very invasive as they form these macrometastases. They are immortal, so meaning that we can transplant this tissue many times, and then we can culture this tumor for months or years if we want to follow. And also, most importantly, they are genomically unstable. So if we take the tissue from these tumoral flies, and we do the squashes, and we label the chromosomes with DAPI. So what we can see here in the wild type, so in flies there are only 4 chromosomes. So there is the 4th chromosome, 2 and 3, and these are the sex chromosome. As you can see in one of the tumors that are the classical tumor, that these are the mutations, typical classical tumor suppressor genes in Drosophila. As you can see here, we have aneuploids. We also can find the tetraploids, also can find the triploids with some extra chromosome, which is the extra chromosome Y and extra chromosome 2nd. And we have the subtetraploids when we have the missing chromosome, which also can recapitulate one of the important characteristics of the human cancers. And these are just the representation of aniline chromosome, just to give you how this invasive, how this can be really, so more than 30 chromosomes can appear in one cell compared to the wild-type cell, which only have 4 chromosomes. So these tumor suppressor genes that I listed here are known to be involved, to be involved in asymmetric cell division. So basically in Drosophila, it's very well known that if there is a failure in asymmetric cell division, there is a cancer in those flies. So this is just a cartoon, just very simplified cartoon, just to show you how the wild-type neuroblast, which is a stem cell, neural stem cell in Drosophila, it can self-renew and have these cell-fate determinants, which are here in red and here in green, which can then asymmetrically divide and give rise to a progeny, which is called the ganglion mother cell. And ganglion mother cell will eventually divide once more and give rise to neurons and glia. And this will form this cluster of the neuroblast, which will exit proliferation in the pupa or adult stage. But if we have of the mutants, some of the mutants that are known to be a self-determinant in this asymmetric cell division, so they are all known to have function in asymmetric cell division of the neuroblast, what happens that this cell will not give rise to the ganglion mother cell, will continuously start to divide, and then what will happen is that we have the continued proliferation with the tumor formation. So this happens in most of the tumors, but only one, which is known as a little 3 malignant brain tumor, until now have not been found to have a role in this asymmetric cell division machinery. So the question is there a tumor formation in the larval brain that can be triggered by defects other than defects the asymmetric cell division, was our question. So just a small introduction to tell you what is this gene. So this gene have been identified as a tumors have been identified as a temperature-sensitive mutant, so meaning that this gene in a temperature which is 17 degrees have a normal wild-type brains, which on the temperature which is 29, as you can see, have many-fold bigger optic globes. So it produces a very aggressive tumors, and this tumor will actually kill the larvae. So they don't pass the larva stage, they just die in the larva stage and never give rise to adults, while if this mutant is growing at a permissive temperature, will give rise to the normal adult fly. And these are just pictures from the 95, when actually this mutant have been described. And in these histological stainings, you can see also, as I already told you, so it has a very overgrown optic globes, but also has a hyperplastic overgrowth in the imaginal discs, so the tissue that will eventually give rise to the wings or antenna or legs in flies. And only, so the important point here is that the malignant tumor, so when we transplant, it will only come from the optic globe, so only from the brain, but it will not come from the from the imaginal disc, which is a hyperplastic tumors. So Drosophila L3-MBT has some conserved domains. It's a proline-rich protein, which has a 3 MBT domain. It has a zinc finger domain because it's a protein binding domain. It has also SPM domain, which is known domain to be present in his homologs, which are belonging to the polycomb group genes. That's why have been described as a polycomb-like gene. And also is known as a nuclear protein and have been described as a transcriptional silencer, although it's a part of the DRAM-MMB repression complex, which is a part of retinoblastoma family. It has 2 retinoblastoma family proteins, which are homologs to the human retinoblastoma and MIB-MOVB complex, and this repression complex represses many of the developmental genes in Drosophila, and also is known that it binds to a core histones. As you can see here in this picture, so we have just a driver for which drives the expression of the cherry MBT, which is expressed in the nucleus of the neuroblasts, and also in the nucleus of the progenitor cells. So MBT domains have a few homologs. So just briefly, the most closest homologs are the polycomb group genes in Drosophila, which is SEM, which is sex compound midleg, and also the SF-MBT. Also, it has a homolog in C. elegans, which is a part of the retinoblastoma E2F family pathway, and also has a 3 homolog in humans, which are also known to be involved in cancer. So in human, L3-MBT has been mapped near to the region which is 20q12, and it's frequently mutated in myeloid leukemias, and also have some of its homologs in humans have a homozygous deletions that have been found in human medulloblastoma. So there is a cancer related, so there is a relation between the cancer and this gene in humans. So although this is a transcription factor, our main questions in this project were basically what genes are regulated by this transcription factor, and actually what is the contribution of these genes for the MBT tumor progression. So although it's a transcription factor, what we did is that we did a genome-wide expression profile, and this just want to tell you what we did actually. So what we did is that we profiled the larval brains from the tumors. So what we have the L3-MBT-TS mutant that is temperature sensitive, so in 29, which gives a very big tumor. So we take the tumoral mass, extract the RNA, and then we do the same for the another allele, which is also known to have a temperature sensitivity. And then we do the same in a transallelic combination. And then just for comparisons, we also take the larval brains from the tumors that are coming from a different origin, which are coming from a Brad tumors that is involved in asymmetric cell division, the machinery, and is just for the comparisons. And our controls are these mutants, but only at the 70 degrees, so in the permissive temperature when they actually have the wild-type brain, so there is no tumor occurring in these ones, and then we have the normal wild-type brains for the comparisons. Then what we do, we take the tissue from the allograft cultures, and then we follow these tumors in the first, fifth, and 10th round of transplantation for the comparisons. For the comparison, we also have tumors that are coming from a different origins, and they are coming also from the allograft cultures from the first, fifth, and 10th, from the different tumors that are coming from a different genetic origin. So when we analyzed all this MBT data, what we get is like 3 very nice clusters. So the first cluster are all our control brains that are clustering here all together, and then we have the transallelic combination of the MBT mutant that is growing at the 29, then we have another allele that is growing also on the 29 in the other one, so all of them which are actually the brain overgrown tumors, and then we have here the allograft cultures which is clustering in as a third cluster. So just to extract the list that is the most important for the AL3 MBT tumoral growth, so we had our strategy how to deal with this huge amount of data. So basically what we did is that we have, for example, TS1 mutant that is growing at the 29 that is tumoral compared to each of the controls which we have. And then we call this comparison A1, B, and C, and so forth. So, and then what we do is that we take the gene list, which is common between the A, B, and C, and take as a common gene list, and then compare to A prime, B prime, and C prime. So this is another tumor, another allele of the MBT compared to all of its controls, and then the same for the transallelic allele, which is compared to all of its control. And then we end up actually with a list that we can know how to deal with this gene list, which is only 151 genes. But this is not the end, so basically what we wanted to know actually if we can distract of those lists, if these 151 genes, if we can take out the genes that are normally involved for the neural development in other tumors that are coming from Drosophila, and then take only the gene list which is only specific for the MBT. So then we end up with only 114 genes. So we have 102 upregulated and 12 were downregulated. So this gene list, this is 102 upregulated genes, is basically what we call the MBT brain tumor signature. So as you can see here in blue, so this you can see that all our controls have downregulated these genes, and in the MBT brain tumors, as you can see here, they are significantly upregulated in all of the tumoral conditions that we had for the MBT brains. And then if we take this gene list and then look at it and the allograft cultures that are coming from the MBT tumors, so the cultures that we actually, when we implant the tumors inside of the host of the abdomen of the fly, they just become more enhanced and in their expression and more significantly different than to controls. And then this can be distinguishedly different from other tumors that are coming from a different genetic origin, like Brad, LGR, Mira, Prospero, or Pins, that you can see they are different, although some of the genes are appearing to be upregulated, but not as here that we have a cluster of all of genes together. So this gene list, most of the genes have not been known to have a known function, but what we know is that 25% of those genes were known to have a germline function. So this was very interesting for us because we were then asking ourselves what actually the genes that are only specific expression have in a germline, what they are doing in the brain tumor ectopically expressed. And this is just to give you a few examples. What are those genes? For example, nanos and zero population growth or female sterile 1b, they are involved in the germline stem cell division. Then we have two thyrodoxins, which are known in the redox reaction, but only in the germline in Drosophila. And then, for example, we have two proteins which are actually involved only in meiosis, and there is nothing known more about them. And then we have a few proteins which are known as PV and aubergine and crimportejas. They are all involved in PV bioRNA biogenesis, where the PV RNAs are the small RNAs that are actually a bit longer than the microRNAs, and they are only until now have been found in mouse in Drosophila, but only specifically expressed in the germline. But not only that, some of the genes that we have found also which doesn't meet our very stringent criteria, we have another genes which are also upregulated in the MBT tumors, and they are actually interacting between themselves. So we have this synaptonemal complex protein gene which interacts together with CONA and hold them, so meaning that actually some structural proteins that are only involved in meiosis are present in the brain tumors actually here. And then we have a bunch of other proteins that are involved in the pvRNA biogenesis, and they are also expressed in the MBT tumors. So the next question was actually if this only happens on the mRNA level, so we wanted to see if these genes are actually translated. So what we did, we have some available antibodies, and then we checked for a number of the proteins, and then we checked by the Western blot and immunohistochemistry. As you can see here in the Western blot, we have these two proteins, which are PV and aubergine, so which are involved in the RNA interference and biogenesis of the small RNAs. As you can see here, we have they are only expressed in the wild-type ovaries as our positive control. Then they are not expressed in the wild-type larval brains, not expressed in the MBT mutant that is growing at the permissive temperature, but expressed only in the tumoral brains that are growing at the restrictive temperature. And just for the comparison, we can see that in the tumors that are coming from a different genetic origin, they are not also expressed. Also here you can see in the neuroblasts, which are here marked, so this is Miranda, which is a neural stem cell marker for neural stem cell in Drosophila. As you can see here, they are, for example, squash, which is only present in red and is present only in the MBT tumors, and also the synaptonemal complex protein gene that is now present in mitotically dividing cells, and this is a protein which is only present in meiosis. So we have a few other proteins that we also checked, but the mRNA levels were highly upregulated, but we couldn't find them actually on the protein levels. So also, although we have found many of the genes that are involved in the pvRNA biogenesis, so the small non-coding RNA biogenesis, we actually wanted to know if these small RNAs are, so if they are actually synthesized in these brain tumors. So we performed a deep sequencing analysis. As you can see here, our positive control is the wild-type ovary. This is our wild-type brain, and this is the MBT brain, which is growing on the temperature, which is a 29. So this is a tumoral brain. As you can see here that many of the pvRNAs are actually synthesized, and in the wild-type brain are basically going to 0 or only at the 10% of their level, which are present in the MBT brain, and of course upregulated in the wild-type ovaries. And also, as you can see here, we have some important microRNAs for Drosophila, which is Drosophila LED7, and also we have Drosophila bantam, which is already known to have a role in the proliferation in Drosophila imaginal discs. So then these similar phenomenon have been also observed in the C. elegans. So this soma to germline transformation have been observed in the longevity mutants and also in the retinoblastoma pathway mutants. So basically what they discovered was that they have the somatic expression of the germline markers. Also, they have the enhanced RNA interference pathway. And also these mutants, although they are longevity mutants, so they have decreased insulin pathway, they are very long-lived. So basically what they were, so this is claimed that the germ cells, the somatic cell can adapt some of the characteristics of the germ cells just to become immortal or to be more stable than itself, like somatic cells. Actually, they also find some of the P granules. P granules are one of the, so these are one of the structural components that are only present in the germline, and they are very important for the germline specification. And also, we haven't found any of these P granules proteins like the mRNAs or also RNA helicases. We haven't found in our expression profile highly upregulated, but still we checked on the protein levels. For example, here we have the VASA, which is the RNA helicases, which is one of the like the master regulator of the germline specification. Although we haven't found the mRNA levels, we found on the protein levels high expression in the MBT brains that were tumoral and that were growing on the temperature, which is a 29, and not present in the wild type. And this also happens if we do the Western blot. As you can see here, we just confirm that this is only present in the wild type ovaries and specifically present only in the MBT brain tumors that are grown at the 29 and not present in the tumors that are coming from a different genetic origin. So actually, we found that VASA expression not only in the brain tissue, so it can be expressed in another tissue, so like the imaginal discs, and also only in our mutant. So although we have found all these genes that are upregulated in our mutant, in our tumors, in the MBT tumors, we were going back to the literature and we wanted to know if so we went back to the literature and then we saw that actually in humans there is a phenomenon which is called a cancer testis antigens, which are normally expressed in the germline, but they are aberrantly expressed in a range type of the human cancers. So these higher levels of the CT antigens have been correlated with poor prognosis, and they are also found in a very advanced tumors. So far, there are more of 100 CT antigens have been identified. There are like around 40 families of those genes. And also, as we have found in our tumoral model, they are frequently co-expressed. So you don't find like one gene expressed and then the bunch of them also become expressed. Also, they are known to be immunogenic, although they are found only in a germline, and they also have potential to be used as a tumor vaccines. And also, more importantly, what we have found that some of the genes that are actually upregulated in our MBT tumor signature, they are actually present in this CT antigens list. So we have some of the homologs like PV, which is known to be present in many of the solid tumors, nanos that is already known to have a role in the metastasis. And then we have the synaptonemal complex gene, which is also present in the high-grade glioblastoma and astrocytomas. And then we have also a VASA protein, which is present in a different retinoblastoma tumors. What in human tumoral biology until now was not known what is the function of these CT antigens. So basically what they know that they are aberrantly ectopically expressed in the human tumors, but their function until now was not known. So the main question in this project was if there is any relevance of the functional relevance of the expression of these germline genes in the brain tumors. So basically what we did is a small screen. So just to explain you how we did is that we have the wild type brains, but we can actually measure the size of those wild type brains. And then if we inject them in an assay for the tumoral growth, as you can see here, in flies, there is no tumor appearing. So it's the percentage of the tumor growth is going to the 0. But then if we have the MBT tumors, so as you can see here, this is like a 5-fold bigger brain, and then if we inject the pieces of those brain, we have 70% of the tumor growth in the allograft cultures. So if we mutate one of the genes that are that we found in our in our signature list, if we mutate them in the background of the MBT, as you can see, for example, one of them were just the first one on our list, we don't have any effect. So basically you can see there is no significant difference between these double mutants and the MBT tumors in the brain size of the MBT. And also if we inject them, there is no significant difference into the tumor growth inside of the allograft cultures. But most importantly, if we mutate one of these genes like PV, VASA, aubergine, or nanos, we have drastically reduced the size of the brain. So just in situ, if we look at these brains, we see that they are becoming as a wild-type brains, and they are significantly similar to the wild-type brains. So they are becoming wild-type-like, and in case of the PV, if we inject these pieces of the brain, we only get a 40% of the tumors appearing, but in the case of VASA, aubergine, and nanos, we go down to 0, so there is no tumor appearing in these double mutants. We also just wanted to check if this can have the effect on the tumors that are coming from a different genetic origin. So basically what we did is we make a double mutants with BRAT, as you can see in this case for BRAT PV, but we did a few more. So there is no effect on the brain tumor growth, so they are significantly similar between the BRAT and BRAT PV double mutant. And if we inject this into the allograft culture as one of our assaying control, we see that there is no difference between the tumoral growth. So the conclusion was that actually the PV, VASA, aubergine, and nanos are contributing to the MBT tumor formation. And this is the most importantly because, as I already told you, in the C. elegans, this soma to germline transformation appears in this retinoblastoma pathway and increased longevity mutants. This appears the same in the humans. In some tumors, when there is expression, ectopic expression of the germline genes that are called the CT antigens, but until now there was no any functional relevance what actually these genes are doing in the human tumors. But what we have found in this Drosophila mutant that we can actually mutate those genes and they can have the tumor suppressing effect. So this already suggests that they can have the tumor suppressing effect in the other species, and also that we can use this Drosophila model as a studying of these CT antigens. So basically that's it. I would like just to thank my supervisor, which for his leadership and guidance through my PhD project. Then I would like to thank Salud, who helped me a lot for with fly genetics, Leire, who provide non-MBT expression profiles, David, who did all the analysis of our gene expression data, all the lab members that were always there for the discussions, and then I would like to thank the genomic facility from the institute that I was working in, and also Spanish ministry that gave me a fellowship for doing my PhD. Also, I would like to thank Andreas for having me here and giving me opportunity to work on the tumor suppressing mechanisms, but in other models, and also the MGC division, which is a great place to be. So thanks for your attention.
Okay, we've got plenty of time for questions. Anna.
So you may have said that, and I may have missed it, but you have in your final functional series of experiments to look at the function of the germline-associated antigens, antigen-encoding genes induced mutation-induced tumors. You had one to show that mutation of BRAT was not affected by mutation of PV, I think.
Yes.
So do you think that they are functioning in completely independent pathways, or do you think that they could be in the same pathway, but mutation essentially is dominant over PB mutation. What's your thought on that?
So what I think actually is that they are completely different pathways, because the BRAF tumors are occurring in the failure in the asymmetric cell division, although BRAF is known as a translational repressor, which is localizing as a cortical protein in the neuroblasts, which we now know that the MBT, the L3 MBT is going completely another way, which is repressing only this germline program in the neuroblasts. So this is what I think, that they are completely independent pathways.
In your 102 genes upregulated by MBT, how many are CT genes?
CT antigens.
CT antigens, how many?
So what we have found until now, the homologs are only 4, but in this very stringent criteria. So this is like only 102 genes.
And
but if we look at the more further expression profile, we found more.
And all 4 of them are absolutely necessary.
Yes, and all 4 of them. We don't know for the C3G, for example, because I didn't do the double mutants for the C3G, but all 3 of them are absolutely necessary for the MBT tumor growth.
Claire?
You mentioned the duloblastoma, so that and the other embryonal tumour in humans, neuroblastoma. Have you compared your gene signature against established gene signatures for those 2 tumour types?
Yeah, actually what we did is this complicated bioinformatics analysis that you can pass your data and see what is the most similar to it, and it appears that it's most similar to neuroblastoma and glioblastoma, our signatures.
Actually, out of the others, are there any that would have the same function? That is, if you make them knocked out, if you knock them out, do they also prevent the growth of the tumor?
So in humans, what they did, for example, is that they culture the gastric tumors, and if they mutate PV, so what they see that these cells are not further fastly proliferating cells, but there is no other of that. There is no any functional relevance, which I know so far have been done.
The question was, out of the 98 others, which are not these CT antigens, are there any that, if you knock them out, that would prevent the growth of the tumor?
I haven't done that. So we were not really interested, although because most of them, the function was unknown. So we were not really interested in doing the screen for the others, but it would be also interesting to see.
Did you look in any other tumour models to see if the PV and VASA or combinations of those genes are likely to play a role in different tumour models?
So in Drosophila, you mean? Yeah. So actually this, it's, as I showed in the last slide, so in BRAF, we know that they are not playing the role in these tumors. And also when we compare the expression profiles, we actually don't see those genes like highly upregulated. So we just can assume that this is not happening in the other Drosophila tumoral models. It's only in this one.
Ross?
Do you have any idea or hypotheses about what a meiosis-specific gene like SCP, the synaptonemal complex, what could a gene like that be doing?
Yes, this is a very interesting question. So what we think is that, for example, these meiosis genes can involve in the aneuploidy actually. So this would be one of the really interesting experiments to do, to compare the double mutant, for example, for the SCP, which is the synaptonemal complex protein, and then do the analysis of the aneuploidies and see if there is like the genome organization, is genome rearrangements are getting less. So because also in human tumors this gene is highly upregulated, it could be that is producing the aneuploidy to these cells that we think, but there is no evidence still.
Claire?
You also mentioned LET7, so do you know if anyone's looking further into whether LET7 is important or essential in driving those tumours?
No, actually we haven't looked at the LET7. We only look at the bantam, which is known to have, so the microRNA that is known to have role in the stopping proliferation. And then, but we did this double mutant. It's true that the brains are much smaller, but actually it's not really relevant because the bantam itself has very small brains because it's involved in the cell proliferation. So, but no, we didn't look at it.
Are there any examples of tumors that are experimentally induced, but where you get metastasis from the natural site without transplanting into the abdomen?
Okay, so they are known in the larvae. So if you have the RAS mutation, in the background of RAS mutation, you would mutate some of the genes involved in asymmetric cell division, like scribbled or DLG. So actually you can see the metastasis inside of the larvae because you can label them with the GFP, and then we can see in the larvae that they are going in the other places that is not, for example, the brain or the imaginal discs. So it's not only the allograft cultures which have the metastasis.
Well, if there's no more questions, it remains to thank Ana.